Hi, happy Wednesday. Today is Wednesday, April the 1st. Not a huge fan. Never really have loved April Fool's Day, but there really isn't a lot of April Foolsing happening today. It's Wednesday. Let's see. Excitement I have for you guys. Not a lot. We are going to talk about molecular cloning today. I'm going to let a few more of you, I don't say show up because it sounds like we're waiting for you to be late, but we're going to wait for the viewer count to get a little bit more exciting. Guys, I saw a bald eagle today. I was out walking the dog before my first class, and I looked up and there was just one sitting in a tree. I took a picture, but it was really far away, and so it's like a cell phone picture, like when the moon is pretty, and then you looked at it, and it's like this big. But you can tell it's like a brown body with a white head, and it's super cute. Around the corner from my house, my apartment, there is a nature preserve, swampy thing, and there's frequently, I see bald eagles in there. Yesterday, when I was walking the dog before it started raining, I looked out and I was like, is that a bald eagle? And then I thought that I was just having like a weird moment, like, oh yeah, it's a bird. It's definitely a bald eagle. Nope, today it was definitely a bald eagle. They must be here because I think they nest around the corner from my house. So we'll see them for the next couple of months. What an exciting day. Lil's here today. She did not love Gen Chem today, but she's here. We'll see how long she makes it. Let me know my previous, in Gen Chem 1, we had some significant buffering problems. My students fixed it one of two ways. You can change somewhere on your screen the resolution. I know that YouTube is making it to where they give you lower quality videos, but you can up that, which might help. And two, you can basically just refresh the screen and you'll miss like 10 seconds, but hopefully it won't be that bad. Sorry, it's totally Florida and there's a gnat in my office and it's making me crazy. Okay, so today in the slides, there's a couple of things that I want to tell you, like in the slide. In the one announcement slide that I posted a little while ago with the cutest picture of Lil, on next week, we're going to keep talking about molecular techniques. I will send you a Friday update with a little bit more of a plan. But if you look in the module for molecular techniques, we're just going to kind of move down that list about different parts, what they are, what they do, how they do anything. We're going to do that. So you can either watch all the Joe videos at once, you can watch them slowly over time. Should work pretty well. There's a chance, and I don't know for sure, that if you click on those, they will autoplay in Canvas. I don't know that for a fact. The tech rep said that that might happen. So if that works, great. Let me know. If it doesn't, click on the video, open it out in Jove, and just make an account. They're free. There's a lot of cool stuff you can learn in there. So I told you that you would have an assignment due next Monday. So that assignment is a figure analysis. There's a new module called figure analysis. I think it's called figure analysis module, whatever. There is a quiz. So it's a quiz. Now, you just need to hit submit by next Monday. You can look at the figure all you want. Feel free to work in groups. Feel free to work around. Look at the figure and the figure caption. You can also, I'm giving you guys the citation. You can go look at the paper. It is open access. You should be able to access that. What you want to do is to ask questions. So what we've done over time is trained you to skip the figures. And you're like, So if you were to read the Gen Chem 1 textbook, there are all of these boxes that most people, myself included, skip over. What we do is we read the text, the body of the text, and skip the figures. In scientific literature, the figures, it turns out, have so much good stuff in there. That is, so if I'm in a hurry, I will just read the abstract, all of the figure captions, and then read the conclusions paragraph. So because in the figure, it tells you all of their experimental basis. It should tell you what controls they ran for their experiment, what they did, how they did it, and anything else you might want to know. So we're going to start to think about figure analysis as we move forward. So that is due on Monday. It should take you like 35 minutes. I want to be really clear. You do not need to read the paper to do your assignment. There is a PDF link of the figure. That piece alone is how you can answer most of those questions. It's worth 15 points. There will be another one here in a couple weeks. So this is what we're going to start with, and then we can go on from there. If you have questions, you can pop in office hours tonight or Monday before that, or you can send me an email and we can meet in Zoom or somewhere else pretty much, I don't want to say whenever you want, but I'm happy to meet with you if you have questions about how to do that. The questions are pretty simple. They're very straightforward. What is this thing in the figure? What is the control in this experiment? All of that information can be found in the figure caption, which is in that PDF. So do not feel like you have to read the paper. So let me move my Gen Chem notes out of the way. So for the next several lectures, we are going to this expression. I basically mean holding. Sorry, that should have glitched out on both ends, but if it didn't, sorry, I made weird faces. So the thing with molecular techniques, it is the ability for us to conduct biochemical experiments. So it's going to be a little bit different format today. You probably want to have a piece of paper or your iPad to draw on or something else, because I'm going to write on that board. I posted on Canvas it's called cloning handout. It's basically a series of guided notes, somewhat similar to the note pages that I gave you guys for similarity a while back. So molecular techniques, what I'm going to teach you the basis of is how we conduct experiments in a biochemical research group. So we want to be able to take a gene from any place that we care about, any other protein, any other organism, virus, plant, bacteria, people, and then we want to be able to look at it in a different system. So we're going to do something called heterologous expression. And then we need to be able to use several of the techniques or the things that we've seen previously. So I think the chat box is on this side. I don't actually have any idea, but we think it's on this side. So you can use that to write notes to me, You can ask questions. I'm happy to answer them. I know that this form is not fantastic, but it is the best way for us to try to answer these questions in this way. So the thing is, before we talk about this, we need to talk about some supplies. So in order to do a biochemical experiment, you need to select a couple of things. Well, maybe if you don't lay in the middle of class, that would be better. Sorry, dog mom problems. Pick a place, anywhere you want to go. Okay. So in order to conduct these experiments, we need to at least start to think about, we only have the handout. There are no more slides, at least unless something drastically changes for the rest of the semester. But there are things to, there's pages to read and Joe videos for you to look at. So the first things you kind of need to think about are, one, what protein, visible, yeah, do I want to express? Two, where do I want to express it? Vector type. And so then the third question that we want to ask ourselves before we even can do any techniques is, so where do I want out of the vector, and where do I want out of the cell type? So the thing is, as we start to think about this, these three things are going to be different for every environment that you encounter. I'd like to apologize for my dog who's playing with some toys behind the board, and so it's going to make like a hot mess of noise, maybe, but hopefully not. Each experiment that you want to do is likely going to have different answers to all of these. So we're going to talk about what types of proteins we can think about, as well as vector types and cell types. Yeah, sorry. That should say, and hopefully use. I will try to write better. I'm going to erase all of this and basically talk about each one of these independently. So the first thing we want to talk about is, what proteins do I want to express? So in a biochemical experiment, your question, your desire is to ask, what does this protein do? Or how can I make this protein do cool stuff? So we're going to start with question 1. So protein type, the thing with protein types, what it's essentially asking is, where did this protein come from? So we can have options, so it could be bacterial. It could be human. It could come from a plant, etc. So the protein type basically tells us, it gives you the idea or the frame of reference. So in my group, we want to study bacterial proteins. That probably means that when we look at cell type and vector types, which are where and how we're going to do the expression, those need to be compatible. If you want to study a human protein, it probably doesn't make any sense to grow it in a fern, like a plant fern, not like some other type of thing. So it matters where all of these things are. All this is to say, you kind of have to think about, what do you want to do before you start doing stuff? So next we're going to talk about, I'll write this underneath, cell types. So it turns out for cell types, there in Dr. Lane's research group, really think about using. So cell type, still see this. So 1 is E. coli. So a lot of times when we think about E. coli, you might be like, but Dr. Malcolm, usually, so BL21 is a modified strain. This modified strain just allows you to express different types of protein. So this would be for storage. And we're able to fix the buffering, because I don't think there's really any good way to fix it here on my end. Increase your quality. Make your quality higher quality, or you can basically like hit refresh and I won't say anything exciting for the next 10 seconds. If it gets really bad, I can stop and restart the stream, which might work. It might not. Oh, yikes. That's okay, we'll go back. Is it less buffering now or still the same buffering? Oh, good. I realized that if it was buffering, me asking you if it was buffering was not going to help. So, sorry about that. Okay, so we're going to go back up to cell types and kind of walk through this again. I'm really sorry. Today seems to be not be my best tech day. So I'm glad it's looking a little better. So cell types. First one is E. coli. So BL21 is a strain that we can express in. DH5 alpha is a storage strain. So what we're going to find out when we talk about vectors is, so what I want you to think about as we talk about this stuff, all of this information is basically taking all of the material we've learned this semester and applying it. So we know that proteins require a promoter. Promoters are specific for genes. So we still need, no matter what protein we want to express, we need to make sure that the vector system and the cell type are compatible. So in E. coli, we can express, so BL21 will express just about anything if it has the right information. XL10 gold, it can express human proteins. So, but XL10 gold has codon optimization. Do you want to, hold on, I gotta let my dog out. Okay. Sorry, I don't know. It's 5 o'clock. She thinks it's time for snacks or cocktails. I don't know. And it's also, it doesn't matter. The light in here is terrible today. So DH5 alpha is a storage strain, and XL10 gold is a human expression strain due to codon optimization. So this is only one type. So let's think about some other types of cells that we might want to use. So you can we have insect cells. So insect cells would allow you to grow different types of proteins in there. They grow slightly differently. So 3 would be mammalian expression. So when we think about mammalian cells, one thing that we all kind of likely know is that mammalian cells need to be culturable in a lab. I'm not saying that we need to take an arm and try to grow protein in it. I'm saying that maybe if we think about E. coli, we tend to grow that in a flask and it shakes. There are ways that we can do the same with mammalian cells. So some examples of that would be HeLa cells. We can have CHO cells. So CHO cells are Chinese. No one's gonna be able to read that. Hold on. We'll just write hamster ovaries. HeLa cells came from Henrietta Lacks. No. HeLa cells are one particular type. There's also a variety. Immortalized, they can be stored in a freezer. And so the other thing about immortalized cell lines is that they are consistent. So we now have 3 different certain types of plants, not necessarily like the plants out in your yard. There are other types of plants that you could use. So once we have the protein that we care about, So if you want to express a bacterial protein, it probably doesn't make sense to grow it in mammalian cells. They're not really the same. The cell membranes are different. The cell dynamics are different. But if you want to look at a cancerous protein, if you want to study how pancreatic cancer advances, HeLa is a type of mammalian cells. These are just different types. So it matters what you want to look at. And so the thing with all of this is it has to do with the types of questions you want to answer. So the next thing we want to think about is our vector. So I'll give you guys just a second to make sure you've gotten all this down. But the vector is going to be part of our expression system. So, I don't know. Hold on, let me make this a little bit lower so you can see it. So vectors are the ability to express HeLa cells come out of a specific organ and they come out of a specific person. Victoria, I'll email you what it is. I don't remember off the top of my head. But I want to say that they are, you know what? I'm not going to guess on the internet. So HeLa cells are just a specific type of lab strain for the purposes of what we're going to talk about. So vectors are DNA fragment vector. The thing is, it has a bunch of components in it. And so we've seen vector maps before. Yeah, that. Hold on, green didn't show up. So when we talked about PCR a while back, feels like 100 years ago, one of the things we talked about is we want the ability to amplify a specific protein. The amplification of a specific gene is so that we can use it in these types of experiments. So in this case, before we start anything else, the blue piece here is our protein of interest. Later today, we will talk, later, I don't know when we'll get to it, but we will talk about how to insert this blue piece into the black vector. So this is our protein of interest. You might also hear me refer to that as a gene. So when we think about a vector, so this part here is our vector. And so vectors are specific for where you want to express them. So if we think about vectors as just a circular DNA fragment, we know that that's one expression for that. But when we talk about them in terms of molecular techniques, the thing is, this is specific if you want to express in E. coli, if you want to express in rat, if you want to express in plant cells or anything else. So the things that make them more alike or different are the next several components we're going to talk about. So one thing that is super important is going to be a promoter. Now, likely, So Johnny with all the numbers after asks the difference between a vector and a plasmid. There really isn't one. I probably use both words interchangeably. You also can use both words interchangeably. One difference that I tend to think about is a vector is a plasmid with no insert in it. So it would be this without the blue piece in there. You can use both words interchangeably in your mind. As far as I know, there isn't a difference, and I don't ever think of a difference. So I hope that helps. But we have this promoter. So the promoter is all things that we've already considered and thought about. So I think we can see if I write down here. The promoter, good, we can see that, is going to contain the -10 and the -35 regions. So the promoter is the same object that you find in any genome, but is specific for this vector plasmid piece. So a lot of times these promoters also are said to contain locks and keys, or locks. We're going to talk more about that in just a second. So the other part of this, there's one other part that's super important when we think about a vector/plasmid. RBS is a ribosome binding site, and the promoters are the -10, -35. So it contains can you guys hear me now, or I don't know what's happening? So the RBS is the ribosome binding site. So this purple thing here is a selection gene. So we will talk about the lock in a little bit. We'll talk about all three of these components in our vector shortly. So the selection gene allows you to know that your vector is in the new host cells. So one thing that we really want to be able to think about is, how do we know if this piece of DNA has been transformed into our new cells? And we can do that via a selection gene. So unfortunately, whiteboard space is quite at a premium here. Yes. So the promoter contains the -10 and the -35, as well as the ribosome binding sequence. It's not a thought. Different promoters contain what's called a lock. We will talk about that here in just a minute. So you have to have a promoter and a vector. Right now, our vector has the protein of interest already in it. Later, we'll talk about how we get that in there and what aspects you need for that. The other thing we have to have is this purple part, which is called a selection gene. It can express an antibiotic. It could express different types of components. We're going to talk about all three of these parts here in just a minute in more detail. So first, I'm going to erase all this and start over with the promoters. So when we think about promoters, we've seen them before. Promoters, we also call them the parts of genes that tell you It's basically a flag that says a gene is coming. So promoters need to contain all of the DNA or RNA markers for transcription and translation. So that encompasses the -10, the -35, and the Schindel-Garneau. So the other thing, so we have the -10 and the -35, the ribosome binding site or the Schindel-Garneau is for translation. So the other thing that people have decided works really well for promoters is to use promoters out of different organisms. So there are two promoters that I specifically sorry, I can tell that it's saying it's reconnecting. So if it just glitched out for you, that's okay. It glitched out for me too. So what I was saying is these can be from different organisms. So organisms such as other E. coli strains, or sometimes we find them out of phages. So phages are the viruses that infect bacteria. So repressor is often used as a promoter. So the LAC-UV5 is E. coli. IPTG. So if you remember back when we talked about or activation, we talked about how the LAC-UV5 or the LAC repressor actually functions. So the LAC repressor prevents transcription. LAC-UV5 is an example of a promoter, yes. So this one works really well in E. coli. It also doesn't lock because the LAC-UV5 promoter is found in E. coli. So, and why you would want to lock is if you only want your protein expressed at a certain time, you need the ability to lock that door. So an example of a promoter that does have a lock is the T7 promoter. This is from Phage, which we'll call a bacterial virus. So we know that viruses have the ability, they need the ability to rapidly replicate both DNA or RNA depending on their environment. So the T7 promoter is often used as a lock promoter. So in vectors that contain the T7, there is another part of that vector that's used to activate this. So the T7 promoter is a phage promoter. It also contains the -10, the -35, and everything else you would need in order to express proteins. So one thing that is important to note about promoters is they come in the gene. So promoters themselves are part of the genes, okay? No, that's not what I meant. Promoters are part of the vectors that you purchase. So every vector you purchase contains a different set or sets of promoters. So these are just two examples of promoters. Do we have any questions about promoters? So Victoria, Are you asking what could turn on both the T7 and the LAC-UV5? So when we talk about these DNA markers or the RNA markers, it is for the regulation off of this plasmid. So these markers will not change the endogenous or the housekeeping gene expression. This is all about the ability to turn on the expression for the blue gene of interest, which I think that's what you're asking. Is that what you're asking? Yeah, no problem. So next, we're going to talk about our purple box, which is selection. So with selection, our goal is to select for cells with the plasmid. So the idea is if we want to express a protein, if I want to express a protein, I need to know which cells have the ability to express that protein. And we can do that by using a selective agent. So clear. Today, most of us, myself included, use digital music, but a cassette is basically one album on a specific thing, kind of like a CD. One thing that we know about those CDs or cassettes, which totally dies when we think about virtual music, is that if you want to do something else, you have to change the CD or the cassette. So an antibiotic cassette is a gene that either creates an enzyme or other molecule that degrades the enzyme. Okay, that's not gonna work. No one can see me and I look like a creeper. So these cassettes, their function is to allow bacteria to grow, or any cell, in the presence of an antibiotic or other selection molecule. There are a couple other ways that you can do this that it requires the use of something else. So some of the antibiotic cassettes that we might think about would be things like ampicillin, kanamycin, tetracycline. Most of those are not necessarily antibiotics that people take anymore. Some of them used to be. I know that my dog has taken kanamycin. For the normal bacterial infections, they are somewhat resistant to that, but the lab-based strains are not. We'll call these types. Ampicillin, kanamycin, or tetracycline. So these are just some antibiotic types. Did you use an antibiotic cassette in the GFP lab? Yes, there should have been one. It might have been the carbanicillin cassette. In that lab, you did use a selection molecule. If you don't, all bacteria will grow. And there's no way to determine whether or not the cells were or were not likely to have this plasmid. Any other questions about selection? Yeah, so the cassettes, I'll explain it and then I'll write some notes up. Yes, the types are different antibiotic cassettes. So a antibiotic cassette is a single gene or cluster of genes that expresses either one enzyme or a series of molecules that will degrade the antibiotic. So this allows the cells to live if they are grown in the presence of an antibiotic. So the bacteria grows in the presence of an antibiotic. What we learned in chapter 25 is that antibiotics kill bacteria. They kill bacteria by preventing cell division, cell septation, protein function. These enzymes are ways that bacteria can prevent the antibiotic from functioning. So antibiotics, when you acquire antibiotic resistance, it comes in one of two flavors. One, you've modified the antibiotic binding site, which is an evolutionary molecule, an evolution of a specific structure. Two, you've evolved to create an enzyme where part of its job is to degrade that molecule. Because if you degrade the antibiotic by cleaving bonds, you can then go in and then the antibiotic is no longer present. So the selection is merely a gene that encodes for antibiotic resistance. Good questions. Any other questions? So the antibiotic does not kill the cell itself. In this case, so John asked, the antibiotic doesn't kill the cell itself, just the bacteria. In this case, the cell is the bacteria. So for antibiotic selection, we are using a bacteria to now become antibiotic resistant because we've added a gene. So the antibiotic only kills bacteria that do not express this selection gene. Vector comes with a different one. So in my lab, we tend to almost exclusively use ampicillin. I think Dr. Nuckley likes to use a kanamycin resistance. Some of that is, for lack of a better term, whatever it was that your old advisor used. So like lab voodoo gets passed down as you become more and more of an adult scientist. So I use ampicillin because that's what my graduate advisor and my postdoc advisor used. But the other thing is some of the cells have antibiotic resistance and you need to use both of those. So antibiotic resistance can be already in the cell and in the vector. Most of the time that is when you go to the biological store, you buy a vector that already contains a promoter and a selection cassette. So both of those are things that you just kind of already have. What is not present at the, I don't know, Science Walmart is this gene of interest. Shortly, we will talk about how you can put that into your vector. Yeah, we'll talk about that for a little bit today. Other questions? going to talk about is something that I affectionately call the MCS. The MCS stands for the multiple cloning site. So the MCS in a vector that does not have a protein in it, that looks something like this, what color do I need? So the multiple cloning site is essentially a doorway or a series of doorways that we can insert the gene of interest. So the multiple cloning site is a region that contains multiple restriction enzyme sites. So the MCS, or the multiple cloning site, is a region in the DNA in this vector plasmid that contains a series of restriction enzyme sites. Those we can think about kind of like a specific way for you to cut and paste and open this vector and then insert in your blue gene of interest. So there are, in this second, I can think of lots of different ways that you can do this. I'm going to teach you essentially one way. When you go on to graduate school or any postgraduate studies, there are many ways that you can do this. Today, we're really going to just talk about one or two. So the multiple could insert your gene. So in just a second, I'm going to erase all of this so we can talk about how we can open this. It really houses between 5 to 12 different unique restriction enzyme sites. So if we start with two things, we are going to have our, and I'm just going to draw in our multiple cloning site. The thing is, so restriction enzyme sites, we've encountered before several different times. So these are enzyme sites like NOT1, Bagel 2, or others. Each of these is specific, but what we need to be able to do is to do something called a digest. So we're going to think about using So the restriction enzyme digest can create sticky ends or blunt ends. Both of those have the ability to have matching ends. So we've talked about how restriction enzymes only cut in specific regions. In this case, we're really only going to talk about one type of restriction enzymes, class 2, which are the ones that cut within the site. So we need the ability to match our multiple cloning site and the gene. If you remember back to PCR, which we've talked about, and you can amplify a gene out of any source. we can also add on to the component of that. So these purple things, which I know you will not be able to see, the NCS. These are added during PCR. So when you amplify a gene, your primer can contain other DNA segments that will allow you to digest part of this and put them together. So I'm going to give you guys a second to do this, and then I will erase the words so that I can write new words underneath. Sorry, I know the whiteboard stinks. So our steps here are going to be, I'll write this over here. Our plan is to digest the vector and gene from our PCR with the same enzymes. So if we used, likely the dark purple things are the, yes, the dark purple or the light purple on the end of the gene is the restriction sites that you added with your PCR amplification. So likely you would want them to be two different sites. So we'll make this one purple and pink, and this one just purple. In your multiple cloning site, in this case, we are going to add in, we have a pink one, we have a green one, and then we're going to have, there it is, purple. So these are restriction sites. So when we digest, we want to digest with enzymes that correspond to the things that we see over here. So when we digest this vector, CMCS, the gene of interest. Ooh, neither. So the MCS is the multiple cloning site that we find in the vector before we add the gene in. So this is vector. This is vector plus gene. So what we, as soon as I get out of the way, are attempting to do is to insert our gene of interest over here into the multiple cloning site. It's a great question. With the buffering, I'm not shocked that we're having slight miscommunications. So when we digest, with the pink enzyme and the purple enzyme, we are now going to get linear DNA with a pink site on one end and a purple site on the other. So the digest, we've talked about restriction enzyme digest before, where you can basically cut the pieces of DNA. In this case, we took our circular DNA and we cut it to make a linear fragment. thing is, we also cut our gene of interest. So we're going to digest with both the pink enzyme and the purple enzyme, to where at the end we get our gene with the pink end over here and a purple end over here. So this piece is also linear and it has nothing else on it. So restriction enzymes in this step are we're starting to make pieces that kind of look like they could go back together. So if you remember back to, restriction enzymes we've met both as the ability to cut DNA or to be used as a defense molecule. But the other thing is these matching ends, we can think about them a little bit like Legos, give or take. We know that Legos can fit back together. Restriction enzymes cut matching ends that can go back together. So, yeah, I need like a way bigger whiteboard. But we're now going to add gene piece and ligase. So in your tube, you will have gene is a, oh, I can see that, ligation reaction. So a ligation reaction is where we can mix together two pieces of DNA. So in this case, we have matching restriction enzymes, pink and purple, on both ends of the gene of interest and on our linear fragment. And so when we ligate them together using ligase, the ligase that you found when we put DNA strands back together, it looks like we're having pretty severe technical. Okay. I've learned that if you guys are telling me it lags, me asking about it doesn't help you. So for the next couple of minutes, and then we will pick up with our ligation reaction again on Monday. But a ligation reaction uses ligase, which is the DNA or is the protein that can come along and seal nicks in DNA backbones. By using matching restriction enzymes, in this case, pink and purple, because spring, on both of these, they will have matching interactions. And so they will basically fit together like a puzzle piece. So when you ligate these two things together, we're going to ligate our linear gene, which we have snipped the ends off, and our linear vector that still contains the promoter and the restriction and the antibiotic cassette. We can ligate them together using ligase to create this molecule, which is our vector plus gene. So now we've made a method by which we can express yes. So Johnny with numbers asked, you basically just cut out the MCS. Correct. So the multiple cloning site is a small piece of DNA that basically has a series of doors in it. It has different restriction enzyme sites. Based on what you need, you can pick different ones. So in our color-coded land, we have pink, green, and purple. Now, you couldn't necessarily, you do need to make sure that they go in the right order. So you could use pink and purple, pink and green, but you couldn't use green as the 5' end of your gene if you didn't use purple. So green and pink only go together if you have the 5' to 3', like the directionality of your vector matters. But yes, all we're doing is cutting out the MCS and putting something else back in there. So I know that we've covered a lot today. We've done pretty good, I think. So for the next couple of minutes, I'm going to try to answer any questions you guys have before class ends, and then I will hop into office hours. So if you have more questions, we'll do that. I'm also going to take a picture of this for myself so that next week on Monday, when we pick up here, I will try to use the same color scheme. Ryan asks, does an MCS always come with a promoter before it in the vector? Yes. So there are ways that you can damage the promoter, but by and large, you need the promoter to be before the multiple cloning site so that the promoter can express the gene that you're interested in. So typically, if we were to add the MCS, it would end up, or the promoter would be here prior to the multiple cloning site. The multiple cloning site should be after that. And so sometimes you can get other things to express in that region if you don't insert your gene in frame, but we will talk about that next week. What other questions do you guys have today? Yes, so Victoria asked, is the homework the figure analysis? Yes, there is other homework coming, but not this week. This week is the figure analysis due on Monday. Likely you will get homework, like the usual kind, next week, I think. You will have one more homework assignment prior to the final. Good question. Any other questions? So Ryan asked, the restriction enzymes. Okay, we have two questions. The restriction enzymes you use are based on where the promoter is in the vector. So the multiple cloning site, if you're reading from left to right, it's the promoter and then the multiple cloning site. Each vector that you buy from the science store contains a different multiple cloning site. So your multiple cloning site needs to match your gene. One issue that people often have is you can run into the fact, so the green one, if the green one was in the middle of our gene, we couldn't use it because then it would cut the gene. So there are a few other intricate details that for the purposes of Biochem 2, we're just going to pretend like the multiple cloning site contains single cut sites that we do not encounter in the gene. In real life, that could be a problem. So Johnny asks, is the MCS replaced with the gene of interest that contains the cassette? Or is the cassette separate? The cassette is separate. So the purple resistance cassette is buried both in the vector. It is in the vector before, and so it stays in this large linear piece where you add this piece containing the vector already, and technically the promoter is already here as well. That is a great question. So the question is, how do you know if your gene was inserted if the cassette is there the whole time? That is a fantastic question. And I know it feels like I'm blowing you off, but we are going to discuss how you can determine whether or not you have created this versus this first thing on Monday of next week. But briefly, you can go back and use other restriction enzymes to do that. So Victoria asks, you buy a vector with promoter, multiple cloning site, selection site, and modify it after. So the vector that you purchased from the store looks something like this: a promoter, a multiple cloning site, selection, some other stuff in there. And then we're going to modify it by adding into the multiple cloning site. In the late 90s, people would make their own vectors. I don't know of any contemporary scientists who make their own vectors. At this point, most of us purchase things, and the vectors that I'm familiar with are pet vectors, or there's a series of other vectors that are kind of, it matters what company you buy it from, what they're called. Good questions. There is a way, full disclosure, where you can buy this piece already made. You could pay someone to make that for you. I'm on a budget, so like we tend to make those ourselves in my research group. Any other questions today? All right, I'm going to let you guys go so I can hop in office hours. If you have questions, please stop in office hours. I know that this feels very complicated and very difficult, but we're all making it. Miss you guys greatly, and I will see you next Monday. Bye.